report about phylum Chaetognatha, also known as the arrowworms. So the defining characteristics of these organisms are chitinous grasping spines and lateral stabilizing fins. The general characteristics and feeding: they are free-living and marine. They are small in size. The largest chaetognaths can only grow up to 15 centimeters. The diffusion across general body surface is responsible for their respiratory and excretory functions. There is no specialized respiratory and excretory organs, which explains the diffusion through the body surface. They lack a blood circulatory system, and the circulation of coelomic fluid is due to the inner lining of the adult body cavity, which is filled with cilia. The nervous system is fairly complex. So the anterior tissue is composed of several ganglia, and the posterior tissue is composed of a conspicuous ventral ganglia. So the chaetognaths have scattered fan-like clusters of external cilia that are sensitive to vibrations for their prey and warnings against predators. Their principal food consists of copepods and fish larvae, and they appear as nearly transparent, which is helpful since they only lie motionless and they move through the oceanic currents. There are two rows of spines adjacent to the mouth, which are responsible for grasping prey when the chaetognath darts forward. Now, when the chaetognath captures its prey, it only takes about 1/15 of a second, so the whole process is really quick. Now, they have two rows of short teeth on either side of the mouth, which is responsible for holding prey during ingestion and piercing the prey's skin. Now, the chaetognath wear a pair of small eyes, which each contain 5 cup ocelli. Now, these 5 cup ocelli are directed towards different orientations, so the chaetognaths have a pair that are directed downward, so it is possible for them to see their insides. Now, the chaetognaths are known as arrowworms because they are longer than they are thick. They lack appendages, and they have 1 or 2 pairs of lateral fins and a terminal caudal fin, which are also called sea anchors. Now, the lateral fins are responsible for buoyancy when they lie motionless, and a terminal caudal fin is responsible for its forward thrust motion. The chaetognaths possess a thin cuticle that is not molted. They have a body cavity that is compartmentalized, which consists of 2 septa. One septa separates the head from the trunk, and the second septa separates the trunk from the tail. The gut is linear, and it opens in both ways, at the anterior portion and the posterior portion. The digestion is extracellular, and it occurs mainly on the intestines, and they have no distinct stomach. Now, if you see, those are the parts of the chaetognaths, and if you look at the 2 bulging portions here, those are the seminal vesicles. Now, reproduction is, the chaetognaths are simultaneous hermaphrodites, the male gonads maturing earlier than the female gonads. They are capable of self-fertilization, and there is no copulation during cross-fertilization. Their sperm transfer is indirect, and as you saw in the picture, their seminal vesicles are located outside of their bodies. Now, fertilization is internal, but there is no morphologically distinct larvae, so they develop as miniature versions of the adults. Chaetognath lifestyles and behavior. Most species are planktonic, and they are passive, which means that they move through ocean currents. They are capable of vertical migrations, and they cannot swim against any substantial current. They migrate downward during day and swim upward during night. But their migration and the extent of how far they migrate depends upon species. Now, the family Spodelidae is entirely benthic, and it uses adhesive papillae for temporary adjustments to the substratum. There are only about 120 species known. They are important components in oceanic food chains, and they are indicators of herring, which is a species of fish that are important sources of food for other fishes. They are also indicators of other exploitable fish species. They are now the In 1994, bioluminescence was discovered in one species, but it can only be found in depths that are at least 700 meters. Now, if you look at the two pictures, these are the species of Chaetognatha that are bioluminescent. Chaetognatha prey capture. So Tucson et al. Was able to gather some Chaetognaths and decapitated their heads and was able to extract potential toxins. Now they used tissue cultures of mouse nerve cells so that they may bioassay and look for potential neurotoxins. They also used two chemicals, namely ovane and veratridine, which are responsible for the activation of the sodium-potassium pump, which is responsible for the regulation of the ionic concentration of the body. So the neurotoxin blocks sodium channels, and the ovane and veratridine is responsible for the magnification of the destruction of the sodium channel. Now, when there are neurotoxins present in the Chaetognaths, mainly on the heads, the neurotoxin blocks the purpose of the ovane and veratridine. Therefore, the cells are spared. So Tucson et al. Was able to compare between decapitated Chaetognaths and Chaetognaths that had their heads. And they saw that the decapitated Chaetognaths cells were not spared because they were affected by the ovane and veratridine, which causes the cells to quickly swell and die. So, do all Chaetognaths produce neurotoxins? The answer is the phenomenon is widespread based on the research of Tucson et al., wherein they gathered and collected Chaetognaths from benthic and pelagic species from different oceans everywhere. The papillae of the vestibular ridges adjacent to the mouth are the most likely sites of the neurotoxin. Now these are pictures of the oral regions of the Chaetognaths. Now the Chaetognath relationships. Relationship to other animal groups has long been uncertain. The adult body cavity serves as the true coelom, and it is made up of mesodermally derived peritoneum. Now the embryonic coelom arises from the archenteron, and it is enterocoelous. The absence of a ciliated larval stage is present in their life history, and the division of adult body cavity is divided into three distinct compartments, similar to the deuterostomes found in arthropods. Now the musculature bears some resemblance to the phylum Nematoda, wherein both of them possess solely longitudinal muscles. The alternating contraction of ventral and dorsal musculature of trunk and tail is responsible for the Chaetognath's movement. The two sets of antagonizing muscles through hydrostatic skeleton is possessed by the Chaetognaths, and the muscle contractions are sporadic. Now these muscle contractions are what made scientists and researchers doubtful of the relationship between Chaetognaths and nematodes, because nematodes possess the sinusoidal waves, and these waves are more regulatory, while Chaetognaths have randomized wave motions. They are not eutelic, which means they grow through cell divisions and not growth in cell size. There is no unambiguous Chaetognaths that are preserved as fossils, which explains why their relationships between other animals is still uncertain. And that is the end of my report.